So we're going to be talking about genetic drift, specifically founder effect and bottleneck effect. So genetic drift is the phenomenon where random changes impact allele frequencies. So the genetic drift arises when populations are small. The smaller populations have more pronounced changes to allele frequencies than larger populations do. And we use the measure of variance to model genetic drift. So variance is S squared, and it is equal to PQ over 2N, where N is the number of individuals in the population, and thus a large denominator, meaning that that N, that number of individuals in your population is large. So a large denominator gives us a small variance. And so that means that there would be less drift occurring in a larger N, a larger population. We would have less variance, thus less drift. So we don't expect to have very much change from the original population. Whereas with smaller populations, this S squared this variance would be greater, so we expect to see more change in the allele frequencies due to those random changes that are affecting the small populations than we would see in the larger populations. And so, like I said, the two cases of genetic drift that we'll be talking about are the founder effect and the bottleneck effect. So with the founder effect, we start off with a large population. Let's say this is the entire Hawaii population. And then a small group of this bigger population decide to move to another uninhabited island and establish their own new population here. So this is the new island that they're on, and this is their new population. And this is why it's called the founder effect, because they are the founders of this new island that they're on. So they created their own population. And the frequency of the different colors, which represent the different genotypes between these two populations, it's very different between these two. So we see kind of half and half of the purple and the yellow color. But then here in this new population, we see only one purple, and most of the other colors are the yellow color. So they're very different between these two populations. And the new alleles in this population can only contain the alleles from that the small group of those individuals brought to that new island. So it only has the alleles that this group brought to the island, not the alleles in the whole mother population originally. So there's been a change in the allele frequency here. The allele frequencies have changed due to this random change. So this is an example of genetic drift, because there was a random change that happened, and it caused a change in the allele frequencies. So with bottleneck effect, again, you start off with a very large population here, but then a large number of individuals just randomly get removed from the population. They go through this bottleneck effect, and some of them get removed. We no longer see, in this smaller subset, we no longer see the yellow, the green, the orange colors, the purple colors. We only see the red and the blue. So a bunch of them were removed from the population, and it's called the bottleneck effect because, as you see, we start and then we go through a bottleneck. So we have a narrow part, and then we get wider again. So we have large population, small population, large population here. And so as we go through that bottleneck effect, only a few individuals come out or survive. And these few people randomly survive, and they are the ones who are able now to pass down their alleles to the next generation, which is here. So this resulting large population has different allele frequencies than the original mother population that was over here. So there has been a change in the allele frequencies. It was random, so it is also an example of genetic drift.